So we've just spun our cells down, and we're gonna open our centrifuge. We're gonna pull out our cells, and we should see. If you look at how the bottles are placed, we're gonna see a pellet on the outside of that bottle. So if we pull out our Cree, you're gonna see a pellet right back there, and that's a decent sized pellet. Hopefully these ones will be a little bit bigger. So yeah, those again are bigger again because Cree is a little bit toxic. So we're gonna take these bottles over to the sink, and we're gonna empty them out. Actually, we're gonna empty it out right here into the liquid waste. Now, the way I've labeled my bottles, I put the name of the sample on the top, which is not the best practice, because if I take off all my tops and then there's an earthquake and all the bottles fall on the ground, I have no idea what's what. So you just have to be kind of careful. When you're pouring off your liquid waste, you want to pour away from the pellet, so the pellet's up. Just make sure it all comes off, and then grab a paper towel and place it on a towel, and then just place your sample number right next door so you know what it is, and then we repeat for the next few samples. So find that pellet. The pellet is right on the opposite side. I'm going to pour away from it, and you want to watch the liquid waste doesn't get too full. Hopefully this is going to fit. Final one. Pour away from that pellet. And now we have all of our cells, and all of our cells have a ton of protein in them. So this is kind of the point where we would diverge from DNA prep, like a MIDI prep, versus a protein prep that we're going to be doing. So this pellet is very, very sticky, and the protocol tells you to resuspend in PBS or some sort of a buffer. In my experience, it's very hard to resuspend that pellet, so I have a little a trick that I do. So I just recap, and I vortex it a little while before I add the buffers in, and you can just watch what happens to the pellet as they slowly start to just kind of fall apart. So you can see on the left side is kind of a better example. I'm going to add this one in as well. And so that left one is pretty much kind of good to go. Give it a few more seconds here. So I'm going to pull off the left one, and just do these two. Inside of here, and you'll see that our pellet has turned into kind of a liquid-like consistency. And this one looks like the stickiest. So you can see how this pellet is just kind of holding itself together really tightly, so that one needs a little bit more time. And then we can check our 622, that also looks nice and liquidy. Not resuspending. Maybe we should do a cut here.